Imagine a future where we could tailor-make our children, cure devastating diseases, or even create superhumans. Could gene editing make this a reality? Welcome to the cutting edge of science, to a realm that was once the stuff of science fiction, but is now within our grasp. We're talking about gene editing, a revolutionary technology that promises to rewrite the very code of life itself. What if we could eliminate genetic disorders from our lineage? What if we could engineer crops that can thrive in the harshest climates, offering a solution to world hunger? It's an exciting prospect, isn't it? But it's not without its controversies. The power to manipulate the building blocks of existence comes with an immense responsibility. It raises questions that go beyond the scientific and venture into the philosophical and ethical. Should we play God? Where do we draw the line between healing and enhancing? These are questions that have sparked heated debates among bioethicists, scientists, and the public. Some argue that we have a moral obligation to use gene editing to alleviate suffering and improve the human condition. Others caution that tampering with our genetic makeup could have unforeseen consequences and open a Pandora's box of ethical dilemmas. Despite these controversies, one thing is clear. Gene editing holds immense potential. It could transform medicine, agriculture, and a host of other fields. It could, quite literally, change the world as we know it. But how do we unlock this potential? How do we harness the power of gene editing? The answer lies in a remarkable tool. A tool that has been hailed as one of the greatest scientific breakthroughs of the 21st century. This tool is CRISPR, and it could hold the key to our future. It's a future filled with promise and fraught with challenges. A future where we could cure the incurable, feed the unfed, and perhaps even transcend the limits of our biology. But it's also a future that compels us to grapple with profound ethical questions and navigate uncharted moral territory. The key to unlocking this future lies in a tool called CRISPR. CRISPR, which stands for Clustered Regularly Interspaced Short Palindromic Repeats, was first discovered in bacteria in the 1980s. This mouthful of a name refers to a series of DNA sequences found within the genomes of bacteria. These sequences are remnants of past viral infections, a kind of genetic memory that bacteria use as a defense mechanism against future viral attacks. Let's take a step back to the 1980s when scientists were examining a peculiar pattern in the DNA of E. coli. They noticed repeating sequences, like echoes that were separated by seemingly random strings of DNA. It was like finding a pattern in a language they didn't fully understand. These repeating sequences were named CRISPR, and those random strings of DNA in between, they were snippets of viral DNA, a record of past infections. Now you might be wondering, how does a bacterial defense system relate to gene editing? Well, alongside these repeating sequences, scientists discovered associated proteins, specifically one named Cas9. This protein, armed with a snippet of viral DNA, would seek out and cut any matching DNA sequence it encountered. It was, in essence, a molecular pair of scissors guided by a genetic GPS. But the true genius of this system is not just in its ability to cut DNA. When a cell's DNA is cut, it naturally tries to repair itself. And in doing so, it can introduce changes at the site of the cut. This is where scientists saw an opportunity. If they could control where the DNA was cut, they could potentially control the changes that occurred during the repair process. In essence, they could edit the genes of living organisms. The initial applications of CRISPR were modest. Scientists used the system to make small alterations in the DNA of bacteria, yeast, and other simple organisms. But the potential was clear. Here was a tool that could be programmed to target any DNA sequence, opening the door to precise genetic modifications in a wide range of organisms, including humans. The real game changer, however, came in 2012. In 2012, two scientists, Jennifer Doudna and Emmanuel Charpentier, discovered that CRISPR could be used as a gene editing tool. This breakthrough was nothing short of revolutionary, propelling the field of genetic engineering into a new era of possibilities. Before 2012, gene editing was a laborious, time-consuming, and imprecise process. It was akin to trying to fix a watch with a hammer and chisel. But Doudna and Charpentier's discovery changed all that, turning the hammer and chisel into a scalpel, allowing for precise, targeted alterations to the genetic code. They found that CRISPR, which stands for Clustered Regularly Interspaced Short Palindromic Repeats, could be combined with an enzyme called Cas9 to form a pair of molecular scissors. This CRISPR-Cas9 system could be programmed to cut the DNA at a specific location, allowing scientists to precisely remove, add or alter sections of the DNA sequence. This was a game changer. It opened up a world of possibilities, from curing genetic diseases to improving crop yields, and even potentially bringing extinct species back to life. The potential applications of CRISPR are as broad and varied as life itself. Not only did the discovery revolutionize the field, but it also democratized it. The CRISPR-Cas9 system is relatively cheap and easy to use, meaning it's not just limited to high-tech labs. Researchers around the world in fields as diverse as medicine, agriculture, and ecology have been able to harness the power of CRISPR to advance their work. This breakthrough didn't just make waves in the scientific community, it caused a tsunami. It has fueled countless research projects, spurred a race for patents and startup companies, and sparked intense debates about the ethical implications of gene editing. In recognition of their groundbreaking work, Doudna and Charpentier were awarded the Nobel Prize in Chemistry in 2020, a fitting testament to the impact and potential of their discovery. Their work has given us a powerful tool, one that could shape the future of life on Earth in ways we are only just beginning to imagine. This discovery won Doudna and Charpentier the 2020 Nobel Prize in Chemistry. Fast forward to today, and CRISPR technology is being used in exciting and sometimes controversial ways. CRISPR, the revolutionary tool that allows us to edit genes with unprecedented precision, has been making waves in the scientific community and beyond. In the realm of healthcare, CRISPR is opening up a world of possibilities. Scientists are now able to correct genetic defects, potentially curing genetic diseases that were once thought to be incurable. Imagine a world where conditions like cystic fibrosis, muscular dystrophy, and even certain types of cancer could be things of the past. This isn't just some far-off dream, but a reality that is steadily coming into focus. But it's not just about treating diseases. CRISPR also holds the potential for genetic modification. It's a tool that could allow us to design our future. We could potentially enhance human abilities, creating stronger, smarter, or more disease-resistant humans. Sounds like something out of a science fiction movie, doesn't it? Yet, it's a possibility that scientists are seriously considering. On a more everyday level, CRISPR is also being used in agriculture. It's allowing us to create crops that are more resistant to pests and harsh weather conditions, ensuring that we can feed our growing global population. It's even being used to engineer microorganisms, producing biofuels and reducing our dependence on fossil fuels. All this might sound overwhelming, and it's understandable. CRISPR is a powerful tool, one that holds the potential to change our world in profound ways. It's a tool that can rewrite the code of life itself. But with great power comes great responsibility. As we continue to unlock the potential of CRISPR, we must also grapple with the ethical implications and ensure that we're using this technology in a way that benefits all of humanity. It's a delicate balancing act, one that we'll need to navigate carefully as we step into this brave new world of genetic engineering. The potential of CRISPR is breathtaking, but it also raises some serious ethical questions. As we delve into the ethical considerations surrounding this revolutionary technology, it's clear that CRISPR holds a mirror up to our collective values. It compels us to ask ourselves, just because we can do something, does it mean we should? The potential for misuse of CRISPR is a significant concern. While it could be used to eliminate genetic diseases, it could also be exploited to create so-called designer babies, where parents choose their child's traits, such as intelligence, appearance, or athletic ability. This could lead to a new form of eugenics, widening the gap between those who can afford genetic enhancements and those who can't. Moreover, alterations made to the human genome could be passed down to future generations, potentially impacting the course of human evolution. This is a profound responsibility, one that requires a thoughtful, measured approach. The implications for healthcare are equally immense. CRISPR could revolutionize medicine by making personalized treatments a reality. Imagine a world where we could tailor therapies to an individual's genetic makeup, potentially curing diseases that are currently incurable. Yet there are concerns. What if a mistake is made during the editing process? What if an unforeseen side effect emerges years down the line? We must ensure robust safeguards and rigorous testing are in place before widespread use. Lastly, we must consider the societal implications. The power to alter our genes could redefine what it means to be human. It could change our relationship with disease, aging, and even death. It's a brave new world, and we must tread carefully. In conclusion, the ethical landscape of CRISPR is as complex as the science itself. It's a tool of immense power, one that must be handled with great care and respect. It's up to us, as a society, to determine how we wield this power, and to what end. As we stand on the brink of a new era, the future of CRISPR is both exciting and terrifying.